For those that are new to the field, I guess the first question you're going to ask yourself is, what is nanomedicine? So back in 2005, the European Science Foundation broadly defined nanomedicine as the application or use of nano-sized tools for the diagnosis, prevention, as well as treatment of disease. Really, it's used as a tool to gain an increased understanding of disease states. And really, the ultimate goal is to improve the quality of life. So now we fast forward almost a decade after that initial definition. If we looked at FDA's most recent guidance for industry, considering whether or not an FDA-regulated product involves the application of nanotechnology, surprisingly, FDA hasn't really established a regulatory definition for nanomedicine, nanotechnology, or other related nanomaterial terms. But what they do recognize is that nanomedicine or nanotechnology involves some sort of engineering, whereby materials are synthesized or applied within the nanometer range, usually around 1 to 100 nanometers. So a lot of the nanoparticles that you may have heard of in the literature can be made up of different materials. For example, it can be lipid-based, polymeric-based. It can be inorganic materials, maybe mimics of virus particles, and so forth. So we've gone a long way since the beginning of the nanomedicine field. A highly recognized nanomedicine that was approved was back in 1995, Doxil, which is basically a pegylated liposomal formulation of doxorubicin. It was initially approved for treating Kaposi sarcoma in patients with HIV. So now there's a lot of skeptics out there where when they look at this product, what they see is the high price tag. Right, so the major benefit of this liposomal formulation is the reduced cardiotoxicity. Right, there's a lot of studies kind of showing that the therapeutic efficacy is very similar to the non-liposomal formulation. So a lot of people will initially ask themselves, well, if one cycle of Doxil costs around $4,000, while doxorubicin is only $30, is it worth that higher price tag for that reduced cardiotoxicity? So the field of nanomedicine has evolved significantly since the time of Doxil's approval. Right? We've gone beyond reducing simply side effects of drugs. Right? There's a lot of nanoparticles or nanomedicines are now being used for enhancing imaging. We have also developed nanomedicines for a variety of different diagnostic applications. Furthermore, nanomedicines have also been designed specifically to help penetrate a variety of different biological barriers. They have also been functionalized in a way where ligands such as antibodies are attached to nanoparticles to help specifically deliver drugs to specific cell types. So really, nanomedicines have evolved a lot further because a lot of the initial nanomedicines that have been developed are really administered as a parenteral, in other words, as an injection. So when we start exploring new ways of delivering nanomedicines, for example, intranasally to the lungs, intravaginally, and so forth, really we need to now have a deeper understanding of the nanoparticle formulation. We need to understand the various differences in the physiological environments in which the nanoparticles are exposed to. So for example, my research is focused on developing nanotherapeutics that are delivered to the intravaginal environment. The intravaginal tract is actually acidic. It's around pH 4.0. So now we have to take into consideration the nanomedicines that we develop, are they actually stable within this environment? And by developing these new technologies and understanding these new physiological environments, we're actually opening up a wide area of new therapeutic potential. So here's just another image kind of showing you how we develop these implantable devices for prolonged drug delivery. So now that we're in the 21st century, you hear a lot in the media about machine learning, artificial intelligence, right? So really now people are trying to explore ways to merge machine learning with nanomedicine design. And with the whole interest and concern with global climate change, are we able to also develop greener methods of synthesizing nanomedicines? And of course, with the most recent pandemic that we're all going through right now, there's a variety of different vaccines that are under development. A lot of the nucleic-based vaccines, as you can see here, they will require some sort of carrier system, and a lot of them will involve some sort of nanoparticle system to be able to deliver these nucleic acids into cells. So it's been a good past several years. Back in 2017, for example, Vyxeos was approved by Jazz Pharmaceuticals, right? It's a combination delivery of two anti-cancer agents for the treatment of acute myeloid leukemia. Patisiran was huge as well because it is recognized as the first RNAi product that's been approved. Now, this is big for the RNAi field because this really gives hope that future products within this field will hopefully be approved one day as well. More recently, in the past year, Hensify, a product by Nanobiotics, it's basically a hafnium oxide nanoparticle that's been approved for enhancing radiotherapy. And then again, another siRNA product also approved by Alnylam in 2019, Givosiran, was approved for the treatment of acute hepatic porphyria. So really now we're asking ourselves, what is the future direction of nanomedicine? But I think that's what today's conference is really about. It's really allowing us to learn from our peers from around the world, kind of asking ourselves questions, you know, where can we take nanomedicine to the next level, right? What are the new applications of nanomedicine? What are the new nanomaterials? So we're hoping today will be a great learning experience for everyone that's involved. So I wanted to take this opportunity to kind of briefly describe some of my research interests. A lot of the nanomedicines you will hear today, as well as products that have been approved, are more focused on cancer therapy. My research program is focused on a variety of different disease states. We use nanomedicines for enhancing wound healing as well as antibacterial activity. We have developed nanomedicines for targeted delivery specific to immune cells in the case of, for example, HIV prevention. We have also developed nanomedicines which kind of block HIV target cells as a strategy for reducing HIV infection. So we just published an article recently this year evaluating our therapy in non-human primates. And we're also interested in developing dosage forms which will hopefully enhance patient compliance. So by delivering a dosage form, such as in this case, an intravaginal ring, we're hoping to be able to release drug over a 1 month period. This can include the release of nanomedicines from these implantable devices. So today, I will be talking about what's called nanomicrobicides. So microbicides are simply defined as products that are applied vaginally or rectally for the sole purpose of reducing sexually transmitted infections. They're a nice alternative to condoms. The reason for this is because in a lot of countries, due to social cultural factors, women are not able to negotiate condom usage. As a result, women will now turn to alternative methods such as potentially gels, films, or intravaginal rings to discreetly protect themselves from these sexually transmitted infections. Today, I'll be talking specifically with regards to a project whereby we develop a siRNA-based nanomedicine for inducing autophagy and knocking down a receptor called PDGFR as a strategy for reducing chlamydia infection. We will then discuss very briefly the formulation of this nanoparticle into a biodegradable film, so that it's a little bit more easier for patients to administer this product. So chlamydia trachomatis is the most common sexually transmitted bacterial infection in the world. 80% of chlamydial infections are asymptomatic. Typically, if left untreated, it can result in severe effects. For example, tubal infertility, increased risk of cervical carcinoma, and in men, it can actually result in urethritis and epididymitis. And one of the most concerning issues with chlamydial infection is even though it can be treated immediately with antibiotics, unfortunately, chlamydia is one of the types of bacterial infection where studies are starting to appear demonstrating that they are developing drug resistance. And as you know, with any type of drug resistance, eventually we're not going to be able to treat them. And unfortunately, it will become a huge healthcare concern. So really my research project then is to develop an alternative methods to antibiotics in treating chlamydial infection. So just a little bit about chlamydia. Chlamydia is a gram-negative obligate intracellular bacterium, which basically means that it needs to infect cells in order to grow and replicate. It typically preferentially infects epithelial cells of the genital tract. Usually chlamydia exists in two forms, something called an infectious elementary body. As you can see here, right, you have the elementary body. What happens then is the elementary body will then begin to bind and infect epithelial cells. After some time, it will then form what's called the replicative reticulate body. These reticulate bodies will then replicate and grow, and then eventually convert back to the elementary bodies, which will then rupture out of the cells and infect nearby epithelial cells as well as other cells, even the sexual partners. So typically chlamydia binds to epithelial cells via two methods. One is first with the primary receptor called the heparin sulfate receptor. So binding to the receptor is considered reversible, meaning that after it binds, it can potentially still come off the epithelial cells. But there's a secondary receptor, the PDGFR beta. Once the binding to the secondary receptor occurs, it's usually considered irreversible. So platelet-derived growth factor receptor beta, it's been shown to play a role in vascular development. But interestingly, studies have shown that disrupting PDGFR beta, it does not result in any adverse effects. There's actually an FDA-approved inhibitor of PDGFR beta. And again, inhibiting this receptor does not result in anything adverse. So autophagy. Most of you are aware of the common cell death modalities, necrosis and apoptosis, right? There is a third type of cell death called autophagy. So typically, autophagy is involved in a way where they degrade cellular organelles and recycle different proteins, especially in damaged cells. So there are actually three types. There's actually something called macroautophagy, chaperone media autophagy, and microautophagy. For the sake of this presentation, I'll actually be referring to macroautophagy. So the main difference between macroautophagy from the other autophagies is macroautophagy has been identified to play an important role in breaking down infectious particles. So whenever a cell becomes infected, macroautophagy may occur, whereby a phagophore will then, basically it's a membrane that forms around the infectious particle. The infectious particle will then be surrounded and form an autophagosome. The autophagosome will then fuse with a lysosome within the body, and then it will become lysed within the lysosome. So that's how infectious particles are broken down within the cells. So our main objective really is to develop a non-antibiotic alternative for the treatment or even prevention of chlamydia. We're hoping to use a combination approach whereby we knock down PDGFR beta using RNA interference to prevent initial binding of chlamydia to epithelial cells. And if unfortunately, if chlamydia is able to bind to the epithelial cells, we're then hoping to be able to induce autophagy so that way we can eliminate or eradicate the infected cells. So a lot of you are familiar with polyethylenimine or PEI, which is a cationic polymer. It's been used widely as a way of condensing nucleic acids for drug delivery. But unfortunately, a lot of you are also aware that PEI can be cytotoxic. So interestingly, there was a recent study that reported that the cytotoxicity induced by PEI may actually be a result of the cells undergoing autophagy. So we wanted to explore the potential of using PEI's ability to induce autophagy as a way of reducing chlamydial infection. So in order to reduce the cytotoxicity of PEI, we decided to encapsulate it within PLGA-PEG nanoparticles to begin with. So for those that are new to the field, RNAi or RNA interference, as you can see on the right-hand figure, basically the idea is you introduce siRNA, which is basically small double-stranded nucleic acids, into cells. So you're going to need to use some sort of carrier system in order to introduce siRNA into cells. Once the siRNA enters the cells, they're going to bind to something called a RISC complex, or an RNAi-induced silencing complex. Once it binds to this RISC complex, the double-stranded siRNA will separate into a single strand, and one of those strands will then recognize the target mRNA, bind to the mRNA, and then cause cleavage of the target mRNA sequence. So our first step then was to evaluate whether or not our nanoparticles are cytotoxic to epithelial cells, or really determine the maximum concentration that we can use. So as you can see here, after 40 hours of incubation, we noticed that once we hit around 5 milligrams per mil of our nanoparticles, we start to see a significant reduction in cell viability. So now our next step then is to also look at whether or not we can detect autophagy within the cells after treatment. So upon treating our nanoparticle formulations to vaginal epithelial cells, we noticed there was only a significant increase in a protein called LC3B. So this protein is actually synthesized during autophagy, and it becomes localized within autophagosomes. So it is a common marker that's been used to determine the number of autophagosomes that are present. So as you can see here, only the non-silencing PLGA-PEG formulation appears to induce LC3B in this part of the study. But again, this is not indicative of whether or not autophagy is occurring, which we'll show later on. So really our next step now is to look at additional markers of autophagy. So we decided to choose 2 markers that are indicative of the initiation stage of autophagy, which is Becklin-1 and VPS34. So these are involved in vascular sorting as well as autophagosome synthesis. And then we also decided to look at 2 degradation stage markers, which are called TECPR1 and UV-RAG. Okay, so these are shown to be involved in the autophagosome lysosome fusion process. As you can see here, in this figure, we see a significant increase in the degradation stage markers, TECPR1 and UV-RAG, in all of the different treatment groups compared to controls. So interestingly, the only group that we noticed an increase in the initiation stage markers is the one that was treated with the non-silencing nanoparticle formulation. So more importantly, these different markers, they really just tell us whether or not autophagy is occurring, okay? I mean, it really just kind of tells us that, you know, there's something happening with autophagy, right? There's different genes that are being produced. But autophagic flux on the right-hand side here, this is what we really use as a defining stage for autophagic degradation, right? Because even though autophagy is occurring, if it's not actually degrading anything, it's not useful to us. So autophagic flux is a great way of determining whether or not autophagic degradation activity is occurring. And what we notice here is, even though all 3 groups, nanoparticle groups, there's autophagy occurring, there's significantly higher amounts of autophagic flux occurring in those containing the PEI within the formulation. So initial studies suggest then that PEI seems to play a role in promoting degradation. So now the next question you'll probably ask yourself is, well, what about PEI alone? Can PEI alone induce autophagy? So we decided then to look at the PEI alone. The concentration here, 37.4 micrograms per mil PEI. That's the same amount of PEI that was used in the PLGA-PEG nanoformulation. So as you can see here, if we use PEI alone, there was a significant reduction in cell viability, which basically speaks to the toxicity of PEI. So really the maximum amount of PEI that we could use alone would be 1 microgram per mil. So we decided then to evaluate whether or not 1 microgram per mil can actually result in any autophagic flux activity. And as you can see here, there was no significant difference in autophagic flux. So what does that mean then? So it basically means that we definitely need to incorporate PEI, for example, into PLGA-PEG to protect it from its cytotoxic effects, okay, but still able to utilize PEI potentially for its autophagic activity. So now our next step now is to really look at the use of our nanoparticle formulation to knock down PDGFR gene expression. As you can see here, we were able to significantly reduce gene expression by 50% after single treatment. Now, keep in mind, a lot of antibiotic treatments, you're going to need to take antibiotics for several days to even up to a week. So after a single treatment, we were able to knock down 50% in gene expression. We were also able to knock down protein expression by around 43% after a single treatment. Now, most importantly, we want to see whether or not our therapy was able to reduce intracellular production of C. trachomatis, the bacterium that's involved. And as you can see here, in the PEI-containing formulation, as well as the one containing the specific siRNA to PDGFR, we were able to significantly reduce the amount of intracellular chlamydia within the cells. And to better see it, you probably can't really notice it in these fluorescent images. We decided to quantitate the amount. And here, circled in red, is the formulation that contains the PEI as well as the siRNA specific for PDGFR. And as you can see here, compared to the controls, there was a significant reduction. We actually were able to reduce 65% of C. trachomatis after a single treatment within the cells. Now, the next question is, what about, yes?
I think we need to go on to the next speaker. Sorry.
Perfect. No,
but if you could wrap up,
so
that would be great.
I'll wrap it up. So basically, the take-home message then is our formulation appears to be effective in reducing C. trachomatis infection. And then here's just a quick diagram showing the biodegradable film that we formulated and how we were able to then actively deliver our formulation into immune cells, because immune cells have also been shown to be reservoirs for chlamydia. So really, there's a lot that have developed since the time of Doxil approval. So I think the big question now we want to ask ourselves are, is nanomedicine still considered the next big thing? All right, so I'm very excited to moderate the session later on today, where Dr. Marcel Bally and Dr. Kishore Wasan will really debate this question, and hopefully we'll get a clear understanding of where the nanomedicine field is headed. So I just wanted to take this time to thank you, the organizers, as well as my team and funding agencies for funding my research. Thank you, and I apologize for going over the talk.